So it's a pleasure to be invited and to speak at this. Obviously, VHL is deeply in my heart. I did my postdoc fellowship at Dana-Farber with Bill Kalin. And again, I'll mention at the end, I was very fortunate that he was willing to take a chance on a physician science, well, actually a physician who knew no science at the time, but has fortunately gotten to know some science over time. But we're all students and we're all students forever. So I'd like to talk to you about a couple of topics that I think fall under the guise of non-canonical tumor-associated antigens in clear cell renal cell carcinoma. These are my disclosures. I believe none of them are really relevant to this talk. And so the two topics I'd like to present include how PBRM1 inactivation and its relationship to endogenous retroviral expression. And then the topic of retained introns in clear cell renal cell carcinoma. Both of these, I believe, are what I would consider to be tumor-associated antigens that are non-canonical in the sense that many of us think of either SNV, so single nucleotide variant neoantigens, or things like frameshift neoantigens, again, that produce epitopes that can be recognized by the immune system. So I think that that is the danger of doing a virtual talk, I guess. So from your office. So as many of you know, the seminal events in clear cell renal cell carcinoma include, let's see if I can get a laser pointer here. VHL loss, as we all know about and have spoken about in depth. But the second most commonly mutated tumor suppressor gene is PBRM1. And I think we need to give credit both to TCGA, but also to Andy Futrell's group, who I think was the first to describe PBRM1 loss of function mutations in clear cell kidney cancer back in 2011. And so at the time when we began to work on PBRM1 as well as its potential role in immune checkpoint blockade response, there were a number of papers that came out suggesting that loss of function PBRM1 mutations were associated with response to immune checkpoint blockade. I've listed some of the seminal ones here. These are all primarily from Ellie Van Allen's group at Dana-Farber Cancer Institute. Subsequently, however, studies of other cohorts, including some that were prospectively collated from clinical trials, did not appear to show a correlation between loss of PBRM1 mutation, loss of function PBRM1 mutations and response to immune checkpoint blockade. And there may be more at this point. Some of the clinicians in the room could likely fill you in on other relevant articles. And then lastly, there was a publication from Tony Chueri's group as well in cancer immunology research that looked at large cohorts of real-world data from both Dana-Farber as well as Memorial Sloan Kettering. And I put that one in the middle because in the Dana-Farber cohort, there did appear to be association between SWI/SNF mutations, of which PBRM1 is a PBAF SWI/SNF complex member, with response to immune checkpoint blockade. But in the Memorial Sloan Kettering data set, there did not appear to be any clear associations. So that one was a little bit of a tiebreaker, unclear which way the tie is going. In any case, so we saw these data coming out. We were interested in what PBRM1 was doing at the time. And so we began to think about, can we mechanistically perhaps tease out whether there's a mechanistic reason for why PBRM1 mutation might alter immune checkpoint blockade responsiveness. Perhaps more important than those correlative studies that I just showed you was a study from Kai Wupert-Fenning's lab at Dana-Farber as well that came out relatively simultaneously with Ellie Van Allen studies. And in this study, they did a in vitro screen for genes that were associated with immune checkpoint blockade in a B16 melanoma model, so in vitro T-cell killing assays. And what was very prominent was that many of the genes that were associated with enhanced ability of T cells to kill target B16 melanoma cells in vitro were associated with PBAF complex. So here's PBRM1. So the more you are up in this area to the upper left, the the more significant your depletion was in killing B16 melanoma cells. So you also see in red ARID2 and BRD7. And what's quite interesting is, those three complex members of the PBAF complex are actually unique to PBAF. And so here's the canonical SWI/SNF BAF complex, which typically incorporates ARID1A and ARID1B, but the so-called PBAF complex incorporates PBRM1, ARID2, and BRD7. And so what's striking is that those three members fell out quite highly significantly in this CRISPR screen. So if this is all true, then why might PBRM1 inactivation actually be associated with ICI, or immune checkpoint blockade response? And so a number of studies from multiple groups perhaps began to shed some light on this. So this is a paper in Cell Reports from James Shea's group, in which they developed a mouse model that knocked out both, either VHL, PBRM1, or the combination of VHL and PBRM1. And doing pathway analysis as well as network analysis, what they saw was that loss of both VHL and PBRM1 really was promoting upregulation of the JAK-STAT pathway. Taking a figure from the science paper I just showed you, again from Kai Wooper-Fenning's lab, you can see that in isogenic cell lines, isogenic B16 melanoma cell lines, what they saw was that the top regulated pathways that were upregulated upon PBRM1 CRISPR were actually type 1, so interferon alpha, or type 2, interferon gamma response genes. And then finally, from the paper from Ellie Van Allen's lab as well, did a number of gene expression correlates, both from patients as well as from isogenic cell lines, and came up with a very similar pattern of expression where cytokine receptor interactions were upregulated, as well as, again, the IL-6 JAK-STAT pathway. And so one lesson I would give to, it's very sad I'm no longer young, but younger people would be, is know thy neighbor. And so what you'll see as a common theme across the topics I'm about to cover is that by talking to and interacting with people kind of outside your field, one can actually promote very favorable and fruitful collaborations. And so Ben Vincent was a junior faculty member here. And at that time, he was working on endogenous retroviruses. And so we got to talking about my observation, or our observations, and some of his. And so for those of you who don't know much about endogenous retroviruses, these are essentially retroviral elements. They make up a significant amount of the human genome, so up to 9%. Eric Lander and colleagues noted this in the first report of the human genome sequencing. You can see here, these are transposable elements, so-called lines, signs, which make up the majority of them, but retroviral-like elements made up 8%. And so ERVs may, in some respects, stimulate innate immunity through several mechanisms. So because they are double-stranded RNAs, they can promote pattern-like recognition receptors, including things like TLR, but primarily we think is the RIG-I pathway, because again, they're double-stranded RNA viruses. And then obviously some of these endogenous retroviruses or endogenous retroviral elements can actually be translated and presented in the context of MHC, serving again as a potentially non-canonical tumor neoantigen. So work from Kim Rathmell's lab that was in parallel or in complement to work from Ben Vincent's lab showed that there were high expression of ERVs, in particular within clear cell renal cell carcinoma. So this plot is really showing the pan-TCGA cohort, and the y-axis is really showing the level of ERV expression. In this case, they defined it as immunogenic ERVs. And this point way out here is really showing, again, a dramatic enrichment, perhaps, in clear cell kidney cancer. Now, do these associate with the response? Both Kim Rathmell's group as well as Ben Vincent's group had their sort of handpick sounds wrong, but that's the word I'm going to use. Handpicked ERV that did associate with response. So here you can see the pluses and minuses are responder versus non-responder. And so the Rathmel group used the Mayer et al. Annotations for ERVs, and Ben Vincent's group used the Barlegu annotations for ERVs, so they're slightly different. But in any case, these two ERVs, specifically from the ERV3 family, ERV 3.2.1 and 3.2.2, both had higher expression in responders relative to non-responders. And this is HERV 4700, which again had higher expression in responders than non-responders. So there's at least correlative data to suggest that higher HERV expression may correlate with response. And I don't know if Marcin Linehan's still on the line, but you know, much of science is rediscovery, as I like to tell my lab. So it's very important to read literature. And I think Richard Childs from the NCI, who had been doing allogeneic bone marrow transplants back in the late 1990s and early 2000s, had noted a number of aspects that are echoed in our work. So he noted that donor-derived CD8 positive T cells in the peripheral blood were able to target HERV antigens specifically. In 2011, he had nice work showing that VHL loss, and in particular HIF and perhaps even HIF2, were important for the regulation of HERV expression. And then finally, at least in the context of HLA A0201 restricted MHC, there were HERV epitopes that could be shown to have T cell specific responses. So all this data in general, I think, implies that at least in some subset of patients, there may be an immunogenic antigen-driven response to endogenous retroviruses of the HERV-E class in particular. So given those two facts, you know, PBRM1 inactivation had some upregulation of JAK-STAT and interferon signaling, and the data I showed you on the HERVs, we hypothesized that PBRM1 inactivation upregulates HERV expression that might result in enhanced innate and adaptive immunity. And so we generated a logistic regression model for each. We took each HERV and looked at its level in mutated versus non-mutated tumors of the gene, significantly mutated genes from the TCGA. I think you can see on both the top heat map, again, that's showing the minus log 10 p-value. Both VHL and PBRM1 have high and relatively consistent correlation with HERV expression. This is held up in the Emotion 150 data set. And other important genes like SETD2, BAP1, et cetera, were relatively inconsistent, again, across the two data sets. And so we chose to focus on VHL and PBRM1. We recognize that a number of endogenous retroviruses can be expressed from immune cells. And so we wanted to make sure that this was not just secondary to purity. And you can see here, there was no correlation of HERV expression, tumor purity, et cetera. We also saw that this association between PBRM1 loss and HERV upregulation was not found in other tumor types. So we took the pan-TCGA, looked at tumor types that had greater than 5% mutation rate of PBRM1. And again, you see no real consistent pattern of expression. We were curious about whether or not the specific HERV family members were enriched in PBRM1 deficient tumors. And so these are the 11 superfamilies that have been defined by Vergelu et al. And you can see here these dots showing the level of significance are highly expressed or enriched in the HERV ER1 subfamily. We knocked down PBRM1 and UMRC2 cells to show whether or not this was happening at a functional level. Again, everything I showed you thus far is an association. And you can see here again that there are less ERVs that are regulated directly by PBRM1, at least in vitro, but they are significantly upregulated and for the HERV ER1 superfamily as well. So these data show functionally that what we're seeing in our correlative data from multiple data sets, that there is functional relevance to this. And finally, again, based on Richard Childs' work, we tested whether or not HIF1 and HIF2 are necessary for HERV upregulation in the setting of PBRM1 loss. And so in PBRM1 knockdown cells, each of these points is a different HERV. And you can see that in PBRM1 knockdown cells, control cells, they're at this level, but when you knock down HIF by siRNA, either HIF1 or HIF2 in this setting, you see that the majority of them are decreasing in their expression. And this holds up in particularly strongly with the HERV ER1 superfamily. So perhaps PBRM1 loss can actually explain the potential for enhanced immune checkpoint blockade, and particularly through its ability to dysregulate HERV expression. And our data may even show why this association with PBRM1 may be relatively exclusive to clear cell renal cell carcinoma. So HERVs are a distinct class of tumor-associated antigens that are highly expressed in clear cell kidney cancer. PBRM1 inactivation promotes HERV expression, and in particular expression of the HERV ER1 superfamily. And this upregulation is clearly dependent upon HIF. And so if PBRM1 mutations are truly associated with immune checkpoint blockade, then this HERV dysregulation may be a potential mechanism for this. I don't have a lot of time, so I bring this up. I know Eric Yonash and Kim Rathmel had a paper that at face value would contradict what I've shown at a functional level. They saw that PBRM1 loss was associated with checkpoint inhibitor resistance. The main thing I'd point out is that as far as, to my knowledge, these were done in Renka cells, and Renka cells are VHL wild type. And so it's not actually inconsistent with our model, because these Renka cells would not have high levels of HIF and therefore would not necessarily be able to upregulate the ERVs as I'd shown you. And so the second lesson is know thy neighbors. So again, it's nice to have collaborations with one person, but it's also nice to forge collaboration with other people. And so Daniel Dominguez is in pharmacology, and through our faculty meetings, we got to talking about something called retained introns. And so retained introns are really, as they are named, they're introns that are typically spliced out through RNA processing, but in many cases can actually be retained. This is important for development and obviously has to do with the spliceosome. And again, Ellie Van Allen's lab had shown, along with Kwok Wong, that retained introns can be a source of new epitopes in cancer. We were curious if to take a more pan tumor type lens to this. And so we looked at the number of or the percentage of introns that were retained in different tumor types across TCGA. I'm showing you just like the top 10 or so, but you can see here Kirk is clearly on the left and has the highest level of retained introns. We have developed a so-called size score, which is a retained intron score essentially. And you can see, at least in the Kirk data from comparing normal to tumors, that the tumors have a clearly high upregulated retained intron score. These just IGV screenshots. These are just different tumors. And you can see here as our size score, meaning high level retained introns, gets higher, the introns are clearly showing more reads mapping to them. And so we thought to make this perhaps more usable, we developed a classification, which we called either normal-like or retained intron high. A third of the tumors were about retained intron high, and the rest were normal-like. You can see here these this tract is showing the normal kidney samples, and so the normal kidney samples almost uniformly cluster with the normal-like retained intron tumor samples. Retained introns appear to have a worse prognosis for reasons we're not 100% sure about at this time. And so we really wanted to ask, what's functionally regulating the intron retention? And so we did differential gene expression between RI high versus normal-like tumors in the TCGA. You can see here that at least using the hallmark gene signatures, one of the most highly downregulated signaling pathways was mTORC1 signaling. So meaning that when you have low mTORC1 signaling, you have higher retained introns. And you can see here that if you look at the tumors, if you bin tumors into mTOR pathway mutated or not, they do have lower. So mTOR pathway mutated meaning activated, they actually do have lower retained introns, consistent with the notion that low mTORC1 signaling results in higher retained intron levels. And so we wanted to prove this functionally as well. And so this is one of our favorite retained introns. You can see in the TCGA data that for RI high tumors, there's a lot of reads that are piling up in the intron, whereas in either normal-like tumors or in normal kidney, you don't see much at all. We were able to validate that this intron is retained in a number of tumors. And then we functionally tested this. So we treated cells with the mTORC inhibitor everolimus. And you can see here that relative to DMSO, the levels of this retained intron did increase with mTOR inhibition. So we think that retained introns can serve as a rich source of potential neoantigens. Again, remember, these were typically not translated previously, so are gonna look very foreign to your immune system. Across TCGA tumors, the clear cell tumors have the highest level of retained introns, again, based on our score. And that correlates with a poor prognosis. And we're not fully sure, but we think that mTOR signaling is a key signaling node that regulates intron retention, which sort of might make sense because, as you can imagine, as you say undergo nutrient deprivation, RNA splicing is probably a relatively energy and nutrient dependent, highly dependent process. You can imagine wanting to shut that down. So at this time, we think we've uncovered two potential sources that are novel for tumor-associated antigens in clear cell renal cell carcinoma. And they appear to be perhaps, at least with PVRM1, relatively specific to clear cell RCC. Much of science is rediscovery. Again, I would encourage everybody, whenever they think they've discovered something novel, to make sure they look at the literature and see what the founders of science have done before us. And then finally, I think collaboration, particularly with other members that are outside of your comfort zone or community, is both fruitful as well as makes science more fun. And so our future work will obviously try to assess how functionally important some of these tumor-associated antigens I've just shown you are. I'd like to, acknowledge a number of people. Bill Kalin was obviously foundational in my start to science, as was Mike and the rest of the lab. And then Ben, Vincent, Dan Dominguez, important collaborators. This work was primarily done by a number of people in the lab that it's really been a group effort. So I'll leave it at that. Thank you.
Great, fantastic. Thank you, Billy. Okay, I mean, I have so many questions. Olsan has something to ask, but before Olsan, why don't I ask you a quick question? So you mentioned introns. So I'm trying to figure out, so you're suggesting these introns are actually expressed, so not in a classical sense. So normally, the introns are spliced out and out of the cDNA and you never see them, but this is not the case, I'm guessing.
Correct. So we have begun to look at the CPTAC data. So for those of you who don't know, that's essentially proteomic data from TCGA. We do see some evidence of peptides that align to these retained introns. We have not yet gotten far enough to statistically say, RI high ones show more retained introns or not, but in any case, we do see evidence that some of them are expressed.
Okay. Olsan?
Hi guys. I mean, to see both of you talking about science it's like a return to youth. I mean, it's amazing. Billy, thank you very much for your presentation. Fantastic work, I think. Couple of questions. One of them is, have you been able to trace any of these peptides, either the HERVs or the retained introns, to the MHC system? Either immunologically or proteomically. I mean, like IPIN, MHC, and lutein, or I don't know. I mean, I'm thinking now actually on my feet right now. And the second question is, Is it possible, or did you try in your PBRM1 lines to manipulate this pharmacologically to see if it depends on HIF2 alpha by treating them with belzutifan, or, which is, I mean, very important clinical questions, is everybody right now combines belzutifan with anything, including, immune check inhibitors. And the other thing is that, how about JAK-STAT pathway? Is it HIF dependent and not JAK-STAT dependent? I mean, I'm sure you know better than anybody else how to parse these pathways.
Yeah, two good questions, some which I have answers to. So in terms of mapping to MHC, to my knowledge, we have not, nobody that I know of has, in our group certainly has done the IP with mass spec. There are two pieces of evidence that suggest that it might be happening. So the first is that the retained introns have been detected in RiboSeq data from kidney cancer patient tumors, suggesting they are being translated. The second is that in the Vincent JCI paper, they did show tetramer specific T cells to ERV 4700 peptides. So I don't know if I'd consider that maybe better evidence, but so those are the two things for that. We have not looked at HIF2 inhibitors and their ability to affect ERV expression. That's a good one. We should do that.
How about
Great.
JAK-STAT?
So by that question, did you mean?
You said that PBRM1 mutant activates JAK-STAT, and I thought that if that activates JAK-STAT, is it HIF that talks only to the ERVs, or is JAK-STAT that talks to the ERVs?
Got it. I don't know the answer to that. I guess based on, I'd have to look back at the paper for sure, but I think Richard Child's work did implicate HIF2 pretty well functionally. So there might be a role for the JAK-STAT pathway, but I don't think it's a large one. But there may be cooperation at the promoter. I don't know the answer to that. But I should look at his either blood or cancer paper.
Okay, Marston has a question.
Yeah, no, hey, really, really beautiful work on the ERV. It's funny. We've not published this, but in our lab, it's looking to us like ERV is not specific to VHL deficient kidney cancer cell lines anyway. So just an FYI, I'm not sure what that means. I think I might have mentioned that to Bill once, and I think he said, well, it's downstream of HIF or downstream of HIF2. So that would make sense. But it'll be interesting. I love that story. It'll be interesting to follow. I thought you really put that together well. Let me ask you a question also about the retained introns. It's particularly interesting. I mean, I loved what you said. You kind of went over it briefly, though, about the retained introns and nutrient sensing. And now, I personally think we still got a whole lot to learn about the VHL-HIF pathway and VHL function in general, maybe even non-HIF aspects of it. And I think, we know iron sensing can be important there, and whether nutrient sensing or redox sensing is also part of that pathway. I think it's to be determined. But what again, could you elaborate a little bit on that, on the, on your idea on that? I think that's really fascinating. I never would have thought of that in a million years.
So first off, it's complete hand waving. We had just been sort of spitballing as to why this all might make sense. So our notion was that in the setting of either low energy or low nutrient, the ability to splice mRNA is a relatively energy dependent process. And so our notion was that if you have, say, a nutrient, or maybe in this case more energy stress, you might not splice appropriately, which would lead to sort of retained introns rather than splicing them out, if that makes any sense.
Right.
And so, because our data suggests that when you have low mTOR signaling, you have increased retained introns. And so that would be equivalent. The low mTOR signaling would be the equivalent to low energy. But one last thing, I'd love anybody's thoughts on this. So when we look at the association between pathway mutations, if we group TSC1, TSC2, Reb, and mTOR activating mutations together, we see that nice association between mTOR activation and retained introns. However, when we look at PI3 kinase and AKT mutations and PTEN loss, it's actually the opposite. And so I think it's probably a real phenomenon, but I'd love anybody's thoughts. And again, not right now, but please email me if you have thoughts about why there be complete polar opposite patterns of retained introns from having PI3 kinase activation versus having, you know, Raptor, mTOR, TORC1 activation. So.
Yeah, it's a great thought, Billy. Maybe just one quick comment. You know, there's mTORC1 and 2, and there may be some differential regulation there as well to add to the complexity. But great. Thank you for that, Billy.